I'm Alexa Veronica Macias, and today I'm going to be presenting about the human immunodeficiency disease, but most specifically about the HIV type 1 variant, which causes 90% of the cases worldwide. So, first off, the morphology of the HIV type 1 variant are spherical enveloped particles, typically measuring 120 nanometers in diameter with a cone-shaped capsid. Also, they are composed of a lipid membrane enveloped with surface proteins like GB120 and GB41. Usually when we do a HIV-1 variant staining, it's going to involve the staining of the viral components of the virus. Like in this case, it's going to be its RNA for visualizations and analysis. There are two preferable techniques. The first one is immunofluorescence, which uses antibodies to fluorescent dyes to label viral proteins. And the second is flow cytometry, which includes the staining and sorting of variants based on the surface antigen. So all viruses need living cells for its survival. They work by inserting their own genetic material into the cell and altering its metabolic system for reproduction and propagation of more pathogens. For HIV-1 variants, it's going to be the same. What they do is they insert their genetic material in the form of RNA into the living cell and focus on altering its glycolysis process to meet the energy demands of viral replication. Also, they affect its fatty acid synthesis to meet the demand of cholesterol and membrane lipids for viral binding and assembly, and also the pentose phosphate pathway for reverse transcription. So, HIV-1 is a retrovirus that primarily infects CD4 lymphocyte cells. They play an important role in fighting against pathogens that compromise the well-being of the body. It is believed the virus originated in chimpanzees during hunting practices in Central and West Africa that were infected with the simian immunodeficiency virus and were transmitted to humans through contact with infected blood. The main ways to get infected is through fluid exchange like semen or vaginal fluid and also blood. Also, HIV type 1 is the main cause of AIDS, which happens when opportunistic infections will usually surround your organism and are not harmful, take advantage of your weakened immune system, and causes severe infections that compromises your life. There can pass years without getting any symptoms, but when they show, the majority of the times they get confused with the common flu, so you can experience fever, chills, night sweats, muscle and joints pain, sore throat, swollen lymph nodes, among other things. So HIV type 1 enters the host cell primarily through its enveloped glycoprotein, which interacts with the cellular receptor CD4 and co-receptor. This triggers membrane fusion and delivering its genetic material into its cell's plasma. The virulence factors of the virion are crucially its amount of virus in the blood and CD4 plus T cell count, which measures the amount of damage the immune system has received. These two factors are used to assess the severity of the infection. HIV had a big and controversial spread between last century in the 80s and 90s, mostly in the male homosexual community due to sexual contact. This caused initial fear and stigma towards the gay community, but it created awareness and a rapid scientific development. Also, it can be observed in some drug addict communities due to needle sharing, but infections have been in decline since then. HIV type 1 primarily enters the body through mucosal during sexual contact or exposure to infected blood, like I said, needle sharing, and from mother to child during either pregnancy, labor, or breastfeeding. So, since the outbreak a few decades ago, we have understand the way this disease works and attacks. So we, as a society, have implemented measurements of prevention, like protective sex, disposal of used needles, and new medications like PrEP, which stands for pre-exposure prophylaxis, or PEP, which stands for post-exposure prophylaxis, that can prevent getting infected in case you got exposed to the virus. There's also medications that treats the disease in case you already have it, that even though it cannot cure the disease, it can treat the virus to the point of avoiding the development of AIDS and suppression of the viral components, which reduces the risk of transmitting it to others. So that's all for my presentation today, and thank you so much for listening.